Hi everyone, welcome to our Intro to Biotech class. We have had the great opportunity to work very closely with Dr. Singh this semester. We have learned about a variety of procedures commonly used in biotechnology. The first things we learned were how to properly freeze and thaw samples so that they could be saved and used for long periods of time. We have gotten to take a close look at a variety of mammalian cells lines, including Chinese hamster ovary cells, neuroblastoma cells, and breast cancer cells.
The first experiment we performed was the passaging of cells. Cell passaging starts when our cells are at 70% confluency. When we have 70% confluency, we can begin the experiment. We retrieve our cells from our CO2 incubator that keeps our cells at the optimal temperature, which is 37 degrees Celsius. We also warm our media up to 37 degrees Celsius. Once it is warmed, we'll organize and disinfect our biosafety cabinet with all necessary components.
When passaging cells, first we need to get single-cell suspension by removing our media from the flask, washing our cells with PBS, phosphate-buffered saline, and then adding trypsin, an enzyme that aids in the digestion, specifically of proteins like the ones on the bottom of the flask that adhere our cells. We then would put the cells in the incubator for a few minutes to allow the cells to be in single-cell suspension. Once single-cell suspension is achieved, which you can ensure by looking at your cells under the microscope, you can then passage a portion of your cells into a new flask with fresh media in it, and then you have passaged your cells.
We had the opportunity to get hands-on experience isolating RNA and DNA using QIAGEN kits. The QIAGEN DNeasy and RNeasy kits are used to isolate DNA and RNA. We also gained the skill of using QIAGEN Mini Prep Kit to isolate DNA plasmid. These kits make it quick and easy to get high-quality genomic DNA, RNA, and plasmid. Once we get our isolates, the next step is to quantify. We get to do this using the NanoDrop 1, a highly advanced mass spectrometer. We can quantify concentrations from as small a sample as 1 nanogram per microliter.
This QIA Prep Spin Mini Prep Kit can be used to isolate the target plasmid after performing a bacterial transformation.
Bacterial transformation is a critical step in gene cloning, the goal of which is to produce multiple copies of recombinant DNA molecule. These genes carried in plasmids often provide bacteria with genetic advantages such as antibiotic resistance. The transformed cells we selected using an ampicillin plate. Typically, this occurs as the plasmid will express an antibiotic resistance gene to protect the transformed cells and ensure maintenance of the plasmid over time and cell divisions. Many copies of the plasmid are created and passed to daughter bacterial cells in the process. We also had the chance to perform lipofectamine 2000 mediated transfection of our SHSY5Y or neuroblastoma cells. Transfection is the process of deliberately introducing nucleic acids into cells, usually eukaryotic cells. In our case, we worked with mammalian cells, a type of eukaryotic cell. We used various transfection methods such as neon electroporation and lipo-based transfection.
The transfection is successful when lipofectamine 2000 gets nucleic acids through the cellular membrane. You can tell that you have successfully completed transfection by looking at your transfected cells under the microscope to see if they are fluorescent.
This fluorescence observed in our lipo-based transfection is because the transfecting plasmids have genes that code for green fluorescent and yellow fluorescent proteins, or the transfecting antisense oligos are tagged with a fluorescent dye such as Cy3 or Cy5. In addition, our class was introduced to various other research-based projects on epigenetics, cancer, and learned techniques like trizol-based RNA isolation from mammalian cells and converting RNA into cDNA using reverse transcriptase PCR. We use cDNA as a polymerase chain reaction template to amplify genes of interest using gene-specific primer sets. We successfully amplified a housekeeping gene, 18S rRNA, and other genes of interest. Our class is very thankful for the hands-on learning experiences this semester. We are super excited and looking forward to involving our Biotech Center for Therapeutics and Genomics Training's grand research-based projects and taking the upper-level biotech biomanufacturing courses. These are just some of the techniques and experiments my amazing Intro to Biotechnology class has gotten to work on this semester. Also, as the president of Dr. Singh's Biotechnology Innovations Club, we have hosted discussions around current biotechnology innovations, seminars, virtual and hybrid, for the student body, either led by Dr. Singh, myself, a MassBay Biotechnology alumni, or a professional in the biotech industry. And we've also hosted field trips, like this one, to the Museum of Science for the Music of the Genome concert.